Hi, I'm Catherine Zhang from DNA Learning Center. Today we'll continue part 3 for this lab. Here we're trying to understand the genetic foundation for personalized medicine by explore the concept of pharmacogenetics. We're using PTC as example to see how a single nucleotide change in human genome may have an impact on the PTC's bitter tasteability. At part 1 and part 2, we did a lot. Let's have a brief review. We learned that in a family, if dad has all tasting receptors, he can taste the PTC. His genotype is big T, big T. His phenotype is taster. If mom has all non-tasting receptors, her genotype is little t, and her phenotype is non-taster. Their children have both tasting and non-tasting receptors. Their genotype is big T, little t, and their phenotype is taster. We also learned the gene responsible for PTC tasting was identified in 2003, and polymorphism associated with PTC tasting, they're particularly in nucleotide position 145, tasters tends to have letter C there, and non-tasters most of the time will have letter G there. We learn how to study our own genotype by collecting cheek cells, concentrate the cells, extract and purify DNA in part 1. And part 2, we learn how to use PCR technology to amplify a specific piece of DNA and then use restriction analysis to find out if that at a particular location we have a letter C or G there. We use the restriction enzyme HAE3 here. We also learn how to get the gel results, and from there, find out what's the genotype and predict the phenotype. Today, we're going to learn how to taste PTC at home and find out your genotype. Actually, it's pretty easy. I'm going to show you how. Here, I have 2 bottles of testing paper I got from Carolina. I have the link posted on the website. So if you're interested to purchase, you can go use that as a reference. So I bought 2. One is called control paper here. All right, and you see here. Okay, the other one is called PTC test paper. Okay, PTC test paper. And then when you open it up, it has a lot of paper you can use, okay? And so you just need to get 1 piece for each person, all right? Like this. So the way to test is firstly you need to put the control paper on your tongue, like that, for about a minute. Then remove it, close your mouth, try to taste it. Have a feeling what the blank paper tastes like. Then you can use water to wash your mouth. And then next, you're going to get a test paper, PTC test paper, and then put it on your tongue for about 1 minute and remove it, close your mouth, try to taste it. Then you one of the 3 results. Either you taste very bitter and probably you can tell you are taster, very good as your genotype. or you can feel it's a little bit bitter. You are also a taster, but probably your genotype is big T, little t. Or you just taste nothing. You are a non-taster, and your genotype could be little t, little t. All right, that's how we score your phenotype, taster or non-taster. And then from there, you can also sort of predict what is your genotype. Sometimes things may get complicated. You may feel your predicted phenotype doesn't match your real phenotype. When scientists looked into this matter, they found actually it's more complicated than they thought. So we know there are many PTC haplotypes in modern human population. But the most common taster haplotype is PAV. The most common non-taster haplotype is AVI. So whatever we see here still works for majority. All right, now we're going to move to the I should say, a very exciting area. We're going to use computer and do some bioinformatics exercises. Now remember at part 2, we're talking about PCR, talking about the PCR result. And I'm not sure if you noticed or not, the PCR product should be 221 base pair length. How did we get that? I'm going to show you. In the PCR process, we know primer is very important because primer, they serve as a bookmark. They will recognize and bind to that specific piece of DNA in your genome and then make copies of it. So primer sequence is very critical for your PCR product. So here I posted the forward primer and reverse primer sequence here. You need to remember that and copy it, and later on we're going to put it into a database to find out the information of the genes and to calculate the size of the PCR product. All right, for next step, we need to go to the internet. Come to Google page and then type in BLAST, blast. All right, and then search blast. Then at the top of the search result is the blast basic local alignment search tool from NIH. You click on that, you will come to this website built by National Institute. Okay, we're going to use nucleotide blast. Click on this button, and then you come to this page. All right, then we need to copy paste the sequence from the previous slide. Then we paste the primer sequence here in this window. And then we need to get rid of all the non-sequence characters here. All right, and then come down. We choose search set first. We will use the standard database, and we will check the nucleotide collection. At the program selection part, we want to optimize for somewhat similar sequence. And then you hit the blast button. then the job will be sent to NIH, and there the computer program will use your sequence as a query to search all the database, all the sequence saved at their database and find the match. And then you got the result back. And you can see we have a lot of matches, all right? So we see Homo sapiens taste receptor, right? We see chimpanzee TAS2R gene. We see gorilla gene. Okay, so that's a lot of information, all right? And so here, let's say we're interested in human. So we're studying human genome. So that's find Homo sapiens taster TAS2R38 gene, and we want a taster and non-taster, whatever, but we need a complete gene. So somewhere down here, you can see there's one said Homo sapiens PTC beta taste receptor gene, PTC non-taster allele. Okay, complete CDS. And next to it is the Homo sapiens PTC beta taster receptor gene, PTC taster allele, complete CDC. And so we will just get either one to calculate the product, PCR product size. Okay, let's say we choose the non-taster allele. Okay, when you open it up, click on that, then they give you a detailed match. Like in the sequence of Homo sapiens beta taster, PTC beta taster receptor gene, where your query matches the subject. So we can see here, we have both the forward primer and reverse primer matches in the same gene. And then we got 4 numbers from the subject, which matches your query. That number here, 101, 142, 321, 297, are the beginning and end of the match DNA nucleotide on the subject sequence. What you need to do now is to find the biggest number among these 4 numbers, which is 321 here, and then the smallest number among these 4, which is 101. You use the biggest number minus the smallest number. 321 minus 101 is, that's right, 220. And then you need to plus 1 to get the actual PCR product size. So that gives you 221. So that's how you calculate the size of your amplicon. All right, now let's solve the riddle. Which is the normal state, tasting or non-tasting? To solve this problem, we need to compare the sequence of the non-taster gene and taster gene and compare them with, let's say, a gorilla's taster gene and chimpanzees, those kind of primates taster gene sequence, and see which one is more consistent here. Therefore, give us a sense which one is more original, more normal. Okay, I'm going to show you how to do this now. To do this, we need to go to a different website called Bioservers. This is a website built at DNA Learning Center. And when you get here, you see there's 3 servers. We're going to just use the sequence server here today. You can use it as a registered user or enter as a one-time user. We are going to click on the enter first, and then you're coming to this page. And then I'm going to show you what's this for, okay? So this one is a help window. It helps you to navigate and use this server. For first-time user, it could be very helpful. Otherwise, you can just close that window. Okay, now we come back to the NCBI website. Here, remember in the previous step, we used the primary sequences to fish out a bunch of sequence information from the database. And then we're going to get the sequence information and then compare them. Let's find Homo sapiens PTC beta taste receptor gene from the non-taster allele. Okay, click on that. You come to here, but this one doesn't show you all the sequence you're looking for. So you need to find out the sequence ID and click on that link. That will bring you to this page with the full sequence here at the bottom of the page. Then you select all of the sequence and copy it and put it into the sequence server. You hit the button on the top, create sequence. All right, then there's a new window pop up like this. You're going to paste the sequence in this sequence window and give it a name. We'll name it a Homo sapiens non-taster, okay, gene. And then you click on OK. Then what happens here is you see the sequence get into this new page. All right, you just repeat this step, go back to the NCBI page and click on the descriptions and find out the taster allele sequence information here from Homo sapien. Click on that and click on the sequence ID number, okay? And then scroll down back to the bottom of the page and select and copy the whole sequence and hit on the, let's see, make sure you can see it, okay? Hit on this button on the top said create sequence, okay? Then there will come this pop-up window again, and then you just paste the whole sequence there and give it a name. This is a Homo sapiens taster gene. Click on OK. Okay, then you come back to the NCBI website again. We now got all the human DNA sequence information from both non-taster and taster. We need some relatives sequence information, okay? So let's choose, okay, that's how about the gorilla? We find a gorilla, okay? And click on that, and this is gorilla's TAS2R38 gene sequence. To get the full length sequence, you click on the sequence ID number, and then you come to this page, and then you go down and select all the information here and copy it, and then move it to the DNA sequence server. And then we're going to click on the Create Sequence button on the top, and then you will see this pop-up window. And then you're going to paste your sequence there. Again, you're going to put a name there. It's gorilla. Did we spell it right? Let's double check. Make sure nothing wrong. Gorilla, gorilla. All right, then click on OK. And then what do you see here? You see here, you got gorilla sequence as well. All right, keep doing it. Let's get one more. How about bonobo? Okay, let's see where's the bonobo sequence. Okay, click on description, scroll down, find the bonobo. Okay, this is bonobo's TAS2R3B gene. Okay, and we're going to click on the sequence ID and come down, find the full length sequence and select all the sequence here and copy it. Come back to this page, click on Create Sequence, and there you see the pop-up window, and you put it there, and it's a cute name, right? And then click on OK and close the window. Okay, let's see what you see here now. On this page, you have Homo sapiens non-taster gene, Homo sapiens taster gene, you have gorilla gene, you have bonobo's receptor gene. We're going to choose them all and do a quick alignment, multi-sequence alignment by click this compare button. And hopefully this software will align them together. Okay, then you're going to see this new pop-up window here, and the sequence all lined up, and the yellow highlighter highlights the difference there. The first one, you see bonobo has a different letter at this position, but that's irrelevant, not related to our question. So we're going to skip it. And another place, again, bonobo has something different. Okay, what we're interested in is the place where the Homo sapiens, either taster or non-taster genes, will be the same as gorilla or bonobo. That way we can figure out which one is more ancient. And then you see here at this position, the Homo sapiens taster has C, letter C there. Homo sapiens non-taster gene has letter G there, while gorilla and bonobo all have C there. What do you think? Yes, so that means the taster gene, the C, is more original. Therefore, the taster is more normal. The non-taster is the mutation. All right, we solved the riddle. Yay! Okay, now when we zoom out to see the modern population, we found the global variation in sensitivity to beta taster substances PTC among different modern population on Earth. Different group of people, like for example, people from India, 32.8% of them, they are non-tasters. But let's say Central America, there are only 9.9% people population, they are non-tasters. So this gives you a sense how they distribute it. So let's come back to the topic. How single nucleotide polymorphism related to personalized medicine? Or in another way, what is pharmacogenetics here? All right, so we know, the more we know, we learn about human genome, the more things we find out there surprised us. Basically, there was a paper published in Nature 2010 suggested there are at least 15 million single nucleotide polymorphisms have been identified in the human genome. That means in many, many places, different people can have different sequence there. We can make it a good use in medicine to screen drugs. Let's say if we did a clinical trial and found the same drug has efficiency in a certain patients, and those patients, they all share the same haplotype polymorphism, and some patients, they just don't have any improvement after taking this drug. So we see this drug has no efficacy in clinical trials for this certain kind of group of people. And we look into those people's genome, we'll find a certain sites of polymorphism there, haplotype. Then we can predict this medicine will be efficient to a new person or not by looking at this person's genome, find out the haplotype of this particular patient. If this particular patient has the haplotype which fits in the patient with efficiency in clinical trials, we know this medicine, this drug will be good for this particular person. But if this person's genome suggests he or she shares this haplotype with those patients, had no efficiency, drug efficiency in clinical trials, then we know this drug is not good for this particular patient. This is not a fiction, it's still going, it's already going on. Many scientists are working on that. So this is one of the projects we did at MIT, and you can see their screening drug by looking into the genome data. So basically, science is the future, personalized medicine is future, because then we can prescribe the better fit medicine to this specific person based on his the knowledge we know about his genome sequence data, and that will reduce the burden of the healthcare, that will improve the outcome of the patient, and also will make the physician's job easier. Okay, so we came a long way. We finished 3 parts of this lab. And as a summary, I would like to remind you, science evolves from past discoveries. From the story of this PTC polymorphism, we know everything has a long path in science. From identify non-taster PTC from identify PTC non-taster as a recessive trait in human population, to now we know so much about haplotypes of this PTC taster genes. We just keep going, and it's just a testimony to see science evolve from the past discoveries. And science also points to the future uses. Like I mentioned, future medicine is personalized medicine. And so the more we know about our genome sequence, the better we can apply the medicine, practice medicine. And also I want you to figure out the modern biological research merged genetics, biochemistry, comparative studies, and bioinformatics all together. It's not that simple. It moves between experiments in vitro and in silico. So you need to learn a lot to work as a scientist these days. As I mentioned, personal medicine is the future medicine. It's good for the patient, good for the physician, good for the healthcare system. And there's so many good experiments left to do, and they're waiting for you to do it. So if you want to be a scientist working in this biomedical field, come on, jump into the water. The view is beautiful. And I'm thrilled to hear from you. For any comments, you can leave your comments at the YouTube comment area or contact us via email. All right, that's all for today. So long. Bye.